I think my job today was, I think Dr. Dore has really laid the foundation for this in more ways than he knows. My getting started on microbiota was really an outcome of time that I spent in 1993, 1996 with Professor Jean-François Dejeau at INSERM Unit 290 in Paris. And I think a lot of the work that they did was directly influenced by Joël Doré. So my task today is to tell you a little bit about, we've already heard that there is very little information on the composition of the gut microbiota in Indians. And my task today is to review what we do know. Okay, so these slides are here. This is just very, very briefly to review for all of you where the microbes are located in the gut. The stomach has very few, as you know, it's largely due to the acid there. There are, as we go down the intestines, the small intestine to the duodenum, the jejunum, and then the ileum, the numbers keep increasing. And finally, in the colon, we have huge numbers of microbes, most of which are anaerobic, and there are a much smaller number of facultative anaerobes. I'm also going to introduce you to some of the concepts of how the microbiota are analyzed. I realize that this is a mixed audience, and therefore people will, some of you will already know a bit of this, but others might not. Looking at the microbes taxonomically, what has really advanced the field, and this is where I referred to Joël Doré's earlier work, was the identification of targets in the 16S ribosomal RNA gene, which is shown here, which is very, there are many regions in it, some of which are very specific to individual species, while others are conserved through genera, groups, or even domains. The techniques that I have used in my lab have really been to look at numbers using real-time polymerase chain reaction targeted at the 16S ribosomal RNA, and to look at, of course, in situ hybridization, dot plot hybridization, FISH. These are techniques that have been widely used earlier. To look at diversity, we've generally looked at some form of gel electrophoresis, that's TTG or DGG, I'm not going to tell you what they are, and deep sequencing. So when many of the studies that I'm going to review have come from my lab, and so I'm just going to tell you what are the kinds of microbes that we were interested in. We're really interested in these phyla, the Firmicutes, the Bacteroidetes, Actinobacteria, and Proteobacteria. And these were families which we thought were of considerable interest to us because a number of them are of importance to human health. They are important in anaerobic fermentation in the colon. And so the TTG is the other technique that we use. This is temporal temperature gradient electrophoresis, and you'll see these bands here, which are the standards, and in between we'll see the numbers of bands in each patient's or individual specimens. So looking at the gut microbes in Indians, what we found was that, and this comes from adult Indian communities, we actually have the sequences from a whole range of others that we haven't yet got around to fully analyzing or publishing yet. But this essentially shows that Firmicutes here were by far the largest group, followed by Proteobacteria, and then the Bacteroidetes. And if we compare that, what we in our earlier studies used a cloning and sequencing strategy, and if we compare that with a similar cloning and sequencing strategy that was originally published, we find that we have much less Bacteroidetes than these North American subjects. Of course, these are a very limited number that was studied in the Ekberg study. So when we looked at, we've already heard Dr. Dore tell us about the development of the gut microbiota, that the gut is sterile at birth, colonized during birth, and there are differences between breast and bottle-fed babies, and then during weaning, the flora, the composition of the flora converges. So we've looked at a cohort of babies born in our hospital, and we followed them up longitudinally. Essentially what we did was, while they were in hospital, we took samples on day 1 and 2, soon after they were born, and the second day of birth, and they visited at home up to the age of 6 months. And what I would like to show you here is that if we look at the total number of 16S rRNA gene copies in the stool, that right from day 1, you will find that their guts are heavily colonized. They appear as if by magic from somewhere, and then there's huge numbers in the gut already from day 1. And when we looked at patterns in this cohort of children over neonates over a period of time, we find that bifidobacteria tend to increase soon after birth. Bacteroides prevotella increased only around the age of 6 months, as did Clostridium leptum, and these probably are important, are related to changes in the diet, especially the introduction of complementary foods. We were not able to really look at the effects of breastfeeding versus bottle feeding because more than 90% of our babies were exclusively breastfed. And there were differences between children that were born by normal vaginal delivery and those that were delivered by cesarean section. And essentially what happened was that the children that were born by normal vaginal delivery had more lactobacilli, they had more enterococci, and they had more Bacteroides prevotella than the neonates delivered by cesarean section. But by day 4, these differences vanished. So it was very rapidly changed, and so we didn't really identify what the reasons for the change were. That's something that would be interesting. When we look at numbers, we find that essentially of some important bacterial groups, essentially what the gut flora in the Indian infants or neonates is not very different from what you'd expect in a Western neonate. And that's by just looking at genera or groups of bacteria. But when you look at individual species, and this is a TTGE gel looking at bifidobacterial diversity in neonates born in our hospital, if we look at data from the West, from children born in New Zealand and the UK, bifidobacteria that are found in the infant feces are largely B. longum, bifidum, and bifidobacterium catenolatum. All our infants had a single species, and that was Bifidobacterium infantis. Dr. Dore has already referred to the hygiene hypothesis, and in fact it's speculated that the fact that Western infants are more prone to atopy and allergies could be related to the nature of the bacteria colonizing them at the time of birth. What happens during childhood and adolescence? So as these kids go along, and we've looked at them till they reach the age of, so these are individual groups of children, but coming from a single village. So they all actually had very, very similar dietary patterns. And when we look at them, we see that bifidobacteria tend to decrease as we become adults. Bacteroides keep going up. These are log scales, so they're a little deceptive here. Eubacterium rectale and Faecalibacterium prosnitziae, which are major carbohydrate fermenters and producers of short-chain fatty acids, they peak during adolescence. This is from Yogesh Chowdhury's group in Pune, where they looked at two different families, three individuals in each of these, this first family here and second family here. They were joint families living in the same household, eating the same kind of food. And essentially, this was the youngest in that group, the oldest in that group, showing that there is a reduction in firmicutes with age. And again, here in this second family also, reduction in firmicutes with age. But these are the very limited number of subjects. Environment influences the gut bacteria a lot. So you know that in India, at least in southern India, cattle live in close proximity to where people live. And when we looked at children that were coming from the villages, looked at the stool of children from the villages, and compared them with children living in Vellore city, and we looked at healthy rural adults, we found that rural children had this ruminal gut, the cattle ruminal bacterium, which is butyrovibrio fibrous solvents, in their feces. So this close contact probably also influences what kind of bacteria we acquire. We've already heard about the effects of diet on the gut microbiota, so there's a little bit of data relating to Indians. This is what this is looked at, the difference between vegetarians and non-vegetarians in a group of young women living in a hostel and eating the same kind of diet, except for the fact that some ate meat and the others didn't. And so the diets were fairly homogeneous in these groups. And what we actually found, much to our surprise, was that the girls that ate meat had many more carbohydrate fermenting bacteria in their gut. And that actually went along with an increased expression of the butyryl-CoA transferase gene, which is important in fermenting carbohydrate to short-chain fatty acids in these meat-eating young women. Microbiota, we've heard they have a great connection with nutrition, and we've heard about obesity just now. So again, in obese children located in Vellore, when we compared them with children that were not obese, what we found that there was an increase in Faecalibacterium prosnitziae in the stool of these. Again, this is a carbohydrate fermenting bacterium. It increases energy conservation. I'm not saying that this is the sole explanation for why children are obese, but this is just an association, and there have been many other explanations for what links the gut microbiota to obesity. Of course, we still don't know if it's cause or effect here. In these children, in fact, when we asked them, the dietary history was not very different between the obese and the non-obese children. So it looked as if there's just a difference in the gut flora. There have been several other studies, many of them from India, some of them from India, suggesting that, looking at this again, look, the firmicutes to bacteroidetes ratios, they alter during obesity, in obese children. Interestingly, there's a connection between the gut microbiota and your iron status. And when we again looked at a group of young women in the community who were iron deficient, and for those of you in this room, you'll all know that iron deficiency is very common in India, particularly in women. And here we had these two groups of young women that were anemic or normohemic, and essentially their iron intakes were similar. The only difference in the microbiota we found, again, this is not next-gen sequencing, this was by real-time PCR targeting specific groups of bacteria. We found that there were increased the lactobacilli were reduced in the iron deficient girls. So there could be two explanations for it. One is that, so you might wonder what is the connection between microbiota and iron deficiency. So firstly, we all have been taught when we were undergraduates that iron is absorbed only from the duodenum, and that is certainly not true. There are iron transporters in the cecum and in the proximal colon, and their expression can be increased by short-chain fatty acids, and it has been shown in experimental animals at least that by feeding inulin, which is a fermentable carbohydrate, that you can increase lactobacilli and you can increase the expression of iron transporters in the cecum. So the alternative explanation for this is that lactobacilli are very iron dependent, and so one of the reasons that they are lower in concentration in the stools of iron deficient women could be that there was not sufficient iron for them to grow. So again, cause and effect are things that we need to sort out here. What about intestinal disease? Essentially, we think that our normal healthy gut should maintain a balance between the protective species and aggressive species of bacteria, of microbes. I've just given you a representation here. And when this is altered, this is what you call dysbiosis. And we have evidence that there is a dysbiosis in these conditions, for instance, in colon cancer, in diarrhea, atopic disease, and Crohn's disease, some of which I'll show you in a minute. So again, please remember that I'm only reviewing data that are related to Indian gut microbiota, or what we know of gut microbiota in Indians. So I'm not doing a global survey of this. So when we look at children with acute diarrhea in the community, we all know that diarrhea will wash out the gut bacteria, and that certainly is true, but there is a selective reduction of carbohydrate fermenting gut bacteria in acute diarrhea. So we took children that had acute diarrhea in the community and sampled their stool, and again visited them 3 months later to sample the stool again. And what we found, interestingly, I'm not showing you the other data, I'm sorry, what we found was that Bacteroides, Eubacterium rectale, and Faecalibacterium prosnitziae were all reduced during the acute diarrheal illness. Interestingly, when we look at bifidobacterial diversity, the number of species of bifidobacteria which you find in the stool, which, so again, diversity, please remember from Dr. Dore's lecture, diversity equals richness equals health. I mean, a very naive way of putting it, but probably true. And the bifidobacterial diversity is reduced during acute diarrhea. Now, in colon cancer, again, there are alterations in the microbiota, and what we found in our patients was that Enterococcus faecalis was increased, Eubacterium rectale and Faecalibacterium prosnitziae, which are short-chain fatty acid producing bacteria, particularly they produce butyrate, that these were reduced in the feces of patients with colon cancer. And for those of you who are interested in short-chain fatty acids and colon cancer, you'll remember that butyrate is protective in colonic epithelium to prevent cancer. They were, again, in patients with inflammatory bowel disease, we have major shifts in the gut microbiota. This is just to show you that what happens in patients with Crohn's disease. And bifidobacteria are significantly lower in patients with Crohn's disease, whereas Eubacterium rectale and that was somewhat higher in these patients. Again, we've been interested in Clostridium lectum and Faecalibacterium prosnitziae essentially because of their link with with carbohydrate fermentation. And when we look at diversity of C. lectum, again using TTG, we find that the diversity is decreased in patients with Crohn's disease. Again, less diverse, unhealthy gut. And that's just the numbers that are there. So the second bit of my talk, and I don't have a lot of information to share, was the effect of probiotics on the gut microbiota in Indians. So some of our earlier experiments, we devised an in vitro system to look at effects of probiotics on cytokine output, and this is some years ago. So what we did was to take colonic carcinoma cell line stimulate it with pathogens, and that would increase output of interleukin-8, which is a chemokine, attracts immune cells or to the sites of inflammation. When we used Lactobacillus casei, which is a probiotic here, this is IL-8 output by when these cells come in contact with Vibrio. When we added Lactobacillus casei to this mix, it attenuated the interleukin-8 response. And similarly, there were a couple of lactobacilli that we isolated from normal people, from the stools of normal people, that were able to similarly attenuate the interleukin response to Vibrio cholerae in the HT2 cells. So we did a study where we actually took yogurt. You know, there's a lot of probiotic yogurt in the market nowadays. So we made a probiotic yogurt, and we gave it to a group of young women living in a hostel. The idea of doing this was that the rest of their diet would be controlled, because they all go and eat in the mess at a particular time every day, and it was easy to give them the yogurt at that time. So for the first 2 weeks, they got normal yogurt, which we had prepared. For the second bit of it, they had probiotic yogurt, to which we added a probiotic microbe. And again, for 2 weeks, they were back on a normal yogurt, and we collected stools during these 3 phases. What this shows you is that the number of probiotic bacteria in the stool went up. This is just the genus, went up in the stool at the time that we gave it. But when we look overall at the balance of bacteria, there was no difference between these 3 feeding periods. Interestingly, when we looked at secretory IgA output in stool, that increased during the probiotic feeding. Human beta defensin 2 excretion did not increase at that time. So to summarize, I've just summarized whatever data we have on the gut microbiota in Indians using molecular techniques. There is a bit of information available from 50 years ago, which used culture-based techniques. The nomenclature of these bacteria was different then, so I've only used the molecular data that we have. The challenge really is to find strategies which will help us understand what is cause and what is effect. I mean, in disease, you see a lot of changes. You don't know whether that's secondary to dietary restrictions, it's secondary to the effects of medication. So all this will really require studies which are adequately powered and adequately designed to see what is cause and what is effect. And this eventually may allow us to manipulate these microorganisms and lead to better health. Thank you. And finally, I should acknowledge all the people that you see here. The ones I've highlighted are the ones who really did the work. My collaborators or people who helped me in various ways, and the funders. Thank you.